What if aging isn't just something you see in the mirror? What if part of the aging process can actually be seen at the ends of your chromosomes? They're called telomeres. And every time many of your cells divide, those protective structures tend to get a little shorter. Short enough, and eventually the cell gets the message. We're done here. No more dividing. Welcome to cellular senescence. But there's a 4-amino acid peptide called Epitalon that has been shown in human cells to activate telomerase, lengthen telomeres, and extend cellular replicative capacity. And more than 20 years after some of the original research, a 2025 study demonstrated Epithalon-associated telomere lengthening again. But here's where this story gets really interesting, because if you've spent 5 minutes researching Epitalon protocols online, you've probably seen 5 milligrams, 10 milligrams, sometimes even more. And everybody seems very confident about those numbers because apparently the internet has never been confidently wrong about a peptide before. There's just one problem. A lot of the dosing discussion surrounding Epitalon traces back to Russian research involving something called Epithalamin. And Epithalamin and synthetic Epitalon are not the same thing. In fact, one experiment comparing them reported something that completely changed the way I personally think about dosing synthetic Epitalon. We'll get to that, because I'm going to explain why I personally research 500 micrograms, why I research it for 20 days at a time, why I repeat that cycle 4 times per year, and why stopping Epitalon doesn't necessarily mean the biological effects immediately stop with it. And there's another development. In July 2026, Epitalon went before an FDA advisory committee as part of consideration for the 503A bulk list. No, that does not mean Epitalon is FDA approved, but for a peptide that spent decades largely associated with Russian longevity research, formal FDA consideration is still worth paying attention to. So today, we're going deep. Telomeres, Telomerase, cellular senescence, Epitalon versus Epithalamin, the cancer question, my personal dosing strategy, why Epitalon is traditionally cycled, and where the regulatory conversation currently stands. Because this story is considerably more complicated than take this peptide and live forever. If only biology were that easy. What's going on, everybody? I'm Kareem Haskett, a certified biohacking coach and performance and longevity consultant. On this channel, we break down the physiology behind peptides, hormones, performance, recovery, metabolic health, and longevity, so you understand not simply what something does, but why it works. Quickly, before we get into it, if you're wondering where I source the compounds I discuss, I use 3 pathways. Physician-prescribed pharmaceutical compounds through a telehealth platform and 503A compounding pharmacy, bioactive precision peptides, and a trusted research source for compounds unavailable through my telehealth or bioactive platforms. You can access all 3 through my website at kareemoptimize.com. Link is in the description. And as always, this content is for educational purposes only and should never be considered medical advice. Peptides, hormones, prescription therapies, and other compounds should be evaluated with an appropriately qualified healthcare professional based on your individual health status, medications, and goals. And when I discuss my own Epitalon protocol later in this video, that's exactly what it is. My protocol, not a dosing recommendation. Okay, let's talk about why the little caps on your chromosomes have created so much excitement. Inside almost every cell in your body are chromosomes containing your DNA. And at the ends of those chromosomes are repetitive DNA structures called telomeres. The classic analogy is the plastic cap on the end of a shoelace, the aglet. What's crazy is I can never remember what that thing is called until I'm explaining telomeres. Well, without it, eventually the shoelace starts fraying. Telomeres perform an analogous protective function for your chromosomes. There's a problem though. Because of the way DNA replication works, most somatic cells can't completely replicate the extreme ends of their chromosomes. So as those cells repeatedly divide, their telomeres generally become progressively shorter. Eventually, critically short or dysfunctional telomeres can activate a DNA damage response. The cell may enter senescence. Essentially, they're saying, I've divided enough, I'm retiring. Unfortunately, senescent cells don't necessarily move to Florida and play golf. They can remain metabolically active and influence the tissue environment around them. And accumulating cellular senescence is one component of biological aging. Now, don't oversimplify this. Aging is not telomeres get short, you get old. There are multiple interconnected hallmarks of aging. Mitochondrial dysfunction, genomic instability, epigenetic alterations, loss of proteostasis, chronic inflammation, cellular senescence, and more. Telomere attrition is one piece of a much larger physiological puzzle, but it's an extremely interesting piece because cells actually have machinery capable of rebuilding telomeres. That machinery is called telomerase. Telomerase is an enzyme complex capable of adding telomeric DNA sequence back into chromosome ends. Certain cells retain substantial telomerase activity because they need continued replicative capacity. Stem cells, germ cells, some immune cells, but telomerase activity is generally low or absent in most adult somatic cells. And this is where Epitalon enters the conversation. Epitalon, also called Epithalon in portions of the literature, is tiny. 4 amino acids: alanine, glutamic acid, aspartic acid, glycine, AEDG. 4 amino acids, that's it. This thing isn't exactly a molecular freight train, but biological signaling isn't necessarily about size, it's about the message. And researchers began finding evidence that Epitalon could influence the machinery controlling telomere maintenance. In 2003, there was an experiment that changed the conversation. Researchers treated telomerase-negative human somatic cells with Epitalon. They reported induction of the catalytic component of telomerase, increased telomerase activity, and elongation of telomeres. Think about what that means. Epitalon isn't physically patching your chromosome like somebody repairing a tire. The interesting part is the signal. The peptide appears capable of influencing cellular machinery involved in maintaining the telomere. And researchers took things further. Aging human fibroblasts were approaching the end of their replicative capacity. Researchers exposed them to Epitalon. Their telomeres elongated. And that's not the part I find most interesting. The cells kept dividing. Epitalon-treated cells completed approximately 10 additional population doublings compared with unrelated or untreated controls. Now, does that mean Epitalon gives you 10 extra rounds of life? No. Please don't start planning your 175th birthday party. These were cells in culture. But mechanistically, that's fascinating. Because now we're not simply asking, did a biomarker move? We're asking, did cellular behavior change? And it did. Now let's fast forward more than 2 decades. In 2025, researchers published new work examining Epitalon in normal human epithelial cells and fibroblasts. And once again, they observed dose-dependent telomere lengthening. In normal cells, this was associated with increased hTERT expression and telomerase activity. That's significant because now we're decades removed from some of the original research and we're still seeing biological activity involving the same pathway. But there's something else in that paper we absolutely need to discuss because the researchers also examined breast cancer cell lines and telomere lengthening occurred there too. The apparent mechanism was different, involving evidence of increased alternative lengthening of telomeres or ALT activity. And that's where this conversation gets considerably more complicated. It seems that there's a telomere paradox. If shorter telomeres contribute to aging, then longer telomeres must always be better, right? Not so fast, my friend. Cancer cells have a logistical problem. If they want to keep dividing indefinitely, they need some way of maintaining their telomeres. Many cancers solve the problem through telomerase, or alternative telomere maintenance pathways. So when we're talking about manipulating telomere biology, we cannot reduce this to short telomeres bad, long telomeres good. That's supplement label biology. Actual biology is context dependent. Does the cancer cell finding prove Epitalon causes cancer? No, that's not what the experiment demonstrated. But does it mean manipulating pathways involved in cellular replicative capacity deserves serious investigation? Absolutely. And that's exactly why I don't believe in turning promising compounds into miracle drugs before the evidence gets there. The biology is exciting, but exciting biology and established long-term human safety are two different things. Now let's talk about what I think may be the most misunderstood part of this entire conversation. Epitalon versus epithalamin. Because people frequently cite historical Russian longevity research, look at the doses, and then carry those numbers directly over to synthetic Epitalon. There's a problem. They're not the same preparation. Epithalamin was a complex peptide preparation derived from animal pineal tissue. Synthetic Epithalon is the defined tetrapeptide, AEDG. That's an important distinction. Imagine somebody saying the historical preparation contained a mixture of pineal peptides and they used X milligrams. Therefore, I need X milligrams of this isolated synthetic 4 amino acid signaling peptide. My question is why? Milligrams tell you mass. They don't automatically tell you biological activity. And there's a study that really drove this distinction home for me. Researchers compare Epitalon with epithalamin in a Drosophila model while examining oxidative processes, and they reported something remarkable. In that particular experimental model, Epithalon demonstrated biological activity at a dose approximately 1,000-fold lower than epithalamin. 1,000-fold. Now, everybody put the calculator down because I already know where somebody is going with this. That does not mean you divide a human epithalamin dose by 1,000 and call that your Epitalon dose. That's not what this experiment established. It was a specific experimental model measuring specific biological effects. You cannot turn that into a universal human potency conversation. What it does demonstrate is the principle I'm interested in. Equal mass does not necessarily mean equal biological activity between epithalamin and synthetic Epitalon. And that's important because if the biological activity of these two preparations can differ that dramatically in an experimental system, why would I automatically assume that a historical milligram protocol for one should become the dosing protocol for the other? I don't. And that brings us to what I personally do. My personal research dose of synthetic Epitalon is 500 micrograms per day. And I use that dose for 20 days. Then I stop. And I repeat that cycle 4 times per year. Now, I want to be extremely precise here. I'm not saying the correct Epitalon dose is 500 micrograms. I'm saying that's the dose and cycling strategy I personally use. There's a major difference. My reasoning starts with what we just discussed. Synthetic Epitalon is a defined 4 amino acid signaling peptide. Historical epithalamin was a complex animal-derived pineal peptide preparation. And experimental research has demonstrated that their biological activity per unit of mass can be dramatically different. So I don't personally see a compelling reason to automatically reproduce a 5 or 10 milligram epithalamin style dose with synthetic Epitalon, simply because those large numbers became part of a peptide tradition. But here's the next question. Why only 20 days? And why do people commonly talk about Epitalon in terms of 10-day cycles, 20-day cycles, once or twice per year? instead of taking it every day indefinitely. Because this is where understanding what a signaling peptide actually does becomes important. Epitalon isn't being approached like hormone replacement, where the objective is maintaining a circulating level of something you're continuously replacing. We're talking about a biological signal. You deliver the signal, cellular machinery responds, gene expression changes, downstream biological processes occur, and those processes don't necessarily stop the moment the last dose leaves your bloodstream. That's important because experimental evidence suggests some Epitalon-associated biological effects can persist after the administration period has ended. In animal research, effects have been observed even weeks after an Epitalon treatment course ended. And the broader pineal peptide research tradition, including human epithalamin research, used repeated treatment courses separated by substantial periods without treatment. Yet physiological changes were still observed longitudinally. That's a completely different model from, if I'm not taking it today, it isn't doing anything today. Think of it this way. If you ring a doorbell and you don't need to keep your finger pressed against the button while somebody walks through the house to answer the door, you already sent the signal. And that's the concept behind the cyclical approach. Historically, you'll commonly see pineal peptide protocols built around approximately 10 to 20 days of administration, often repeated periodically rather than continuously. Some longer-term human epithalamin research used repeated courses across multiple years, rather than administering the compound every day. And that's one reason the peptide community still commonly talks about Epitalon in terms of 10 to 20-day cycles, often once or twice per year. But remember, those larger historical doses and much of that course structure came out of research involving epithalamin and the broader Russian peptide bioregulator program. My approach retains the cyclical signaling concept, but changes the exposure. Instead of taking the larger multi-milligram doses commonly associated with historical protocols, I personally research 500 micrograms of synthetic Epitalon daily for 20 days, 4 times per year. So my strategy is essentially lower dose, 20-day signaling window, stop, allow the downstream biological response to continue, then repeat the cycle periodically. I'm not trying to keep Epitalon circulating continuously. I'm trying to periodically deliver the biological signal. And that's a very important distinction. Could 500 micrograms 4 times per year ultimately prove superior to 5 milligrams twice per year? We don't know. Could twice yearly treatment be completely sufficient? Possibly. Could 1 milligram produce a different response? Possibly. Could 5 milligrams? Possibly. We simply don't have adequate human dose ranging and schedule comparison studies used during synthetic AEDG to answer those questions. So again, 500 micrograms for 20 days, 4 times per year is my protocol. It's based on how I interpret the available biology. The potency distinction between synthetic Epitalon and epithalamin and the concept of periodic signaling rather than continuous exposure, it is not an established optimum human protocol. And until somebody gives me the study comparing 500 micrograms, 1 milligram, 5 milligrams, different cycle lengths, and different annual frequencies, we don't get to pretend science has answered that question, because more isn't automatically better. And with the signaling peptide, the objective isn't to see how many milligrams you can fit into a syringe. The objective is the signal. And there's another reason I think the conversation around Epitalon gets too narrow. Everybody wants to talk about telomeres, but this entire story started with the pineal gland. And that opens another major longevity pathway: circadian biology. The pineal gland plays a central role in melatonin production and synchronization of our sleep-wake cycle Research involving pineal peptides has explored their relationship with melatonin secretion and age-related circadian regulation. And that matters because longevity isn't simply how many more times can a cell divide. It's also how well does the organism regulate itself. Sleep, hormonal rhythms, metabolic timing, recovery, mitochondrial function, circadian synchronization. So I don't think Epithalon should simply be thought of as the telomere peptide. Its potential biological relevance is broader than that. And now we get to what happened in 2026. Because Epithalon recently entered a regulatory conversation in the United States that I think is worth paying attention to. In July 2026, Epitolon freebase and Epitolon acetate were considered by the FDA's Pharmacy Compounding Advisory Committee in connection with potential inclusion in the 503A bulk list. The nominated clinical use was insomnia. Now let me make something crystal clear. This does not mean Epitolon is FDA approved. It isn't. And it doesn't mean FDA decided Epithalon works for longevity. That's not what this process was about. As a matter of fact, FDA staff recommended against placing it on the list. An advisory committee consideration itself is not drug approval. So why am I mentioning it? Because I still think it's an important milestone. We're talking about a compound that spent decades primarily associated with Russian peptide research, and it has now entered formal regulatory evaluation in the United States. That's meaningful. Not approval, not validation of every claim made about Epithalon, but it's movement. And when compounds in this space begin receiving formal regulatory scrutiny, I pay attention. Because sometimes the first step toward better evidence isn't approval. It's getting serious enough that somebody finally starts asking the right questions. So how do you know if any of this is working? This is one of my biggest issues with longevity protocols. You inject Epithalon tonight, wake up tomorrow, look in the mirror, and congratulations, you're 1 day older. This isn't caffeine. You're not waiting 30 minutes and saying, oh yeah, telomeres are definitely hitting. If you're trying to influence long-term biological aging, the measurement strategy has to match the intervention. That means looking at trends over time. Potentially, telomere length, epigenetic age, metabolic markers, inflammatory markers, sleep, circadian patterns, cardiovascular performance, body composition, recovery, strength, and overall physiological performance. And remember, telomere age and epigenetic age aren't the same thing. They're examining different components of biology. I don't care about chasing one sexy biological age number. I care about trajectory. Where did you start? What changed? What intervention occurred? Did the biomarker change? Did performance change? Did health change? Can the result be reproduced? Because data without context is just another number on an app. So now, let's get to the question everybody wants answered. Does Epithalon reverse aging? We cannot responsibly say that. We don't have the human evidence necessary to make that claim. But here's what we can say. Synthetic Epithalon has demonstrated biological effects involving telomerase and telomere length in human cellular models. Earlier experiments reported increased replicative capacity in aging cells. Most recent research has again demonstrated telomere length effects. There's historical research surrounding pineal peptides and aging physiology, and now Epithalon has received formal consideration within the U.S. regulatory system. That's enough for me to say this peptide deserves serious research. But there's one study I really want. Defined synthetic AEDG, large human population, multiple dosing groups, randomized, controlled, long-term follow-up, telomere measurements, epigenetic aging, metabolic outcomes, sleep outcomes, cancer surveillance, and comprehensive safety data. Give me that study. Because then we're no longer asking whether Epitalon produces interesting cellular signals. We're asking a question that actually matters. Do those signals translate into meaningful improvements in human health span? So what are the key takeaways? 1. Telomere attrition is one component of biological aging, not aging itself. 2. Telomerase provides cells with a mechanism capable of maintaining and extending telomeres. 3. Synthetic Epithalon has demonstrated the ability to influence telomerase activity and telomere length in human cellular models. 4. Epitalon and Epithalamin are not interchangeable preparations. 5. Experimental evidence demonstrates that their biological activity per unit of mass can differ dramatically, which is why I question simply copying historical Epithalamin milligram protocols for synthetic Epitalon. 6. My personal protocol is 500 micrograms per day for 20 days, 4 times per year. That's based on my interpretation of the synthetic peptides biological activity and the historical concept of short signaling courses. It is not an established optimal dose or dosing schedule. 7. The reason for cycling matters. The administration period may end, while downstream biological effects can persist beyond that dosing window. You're delivering a signal, not necessarily trying to maintain continuous exposure. And finally, 8. Longer telomeres don't automatically equal longer life. If they did, there will already be some 400-year-old guy on YouTube selling a $97 telomere masterclass. Longevity is bigger than one biomarker, and it's definitely bigger than one peptide. It's mitochondrial function, metabolic efficiency, structural integrity, cardiovascular fitness, strength, sleep, nutrition, hormonal health, cognitive function, and cellular regulation. Because the goal isn't simply to add more years. The goal is to preserve the physiological capacity to perform during those years. And if you found this breakdown useful, hit the like button, subscribe to the channel, and tell me in the comments, were you already aware of the difference between Epitalon and Epithalamin, or were you treating those historical dosing protocols as though they were the same compound? And if you want to learn more about the compounds and strategies I discuss, or you want me to build you a free personalized protocol based on your specific goals, head over to KareemOptimize.com. Everything starts there. The goal isn't more compounds. It's understanding the right tools for the right objective and the right strategy. I'm Kareem Haskett. Stay informed, stay intentional, keep optimizing.